Hello friends, how are you? I hope you all are safe at home. Stay home, stay safe. I am again back with few topics of molecular basis of inheritance part 3 of 12th standard. Today's topics for discussion are the search for genetic material and biochemical characterization of transforming principle. The first topic of today's online session is the search for genetic material, that is also called as transforming principle. This principle was given by Frederick Griffith in the year 1928. His experimental material was Streptococcus pneumoniae, a bacteria that causes pneumonia. During his experiment, he found that there was a miraculous transformation in the bacteria by changing the physical form. So let us see what he did in his experiments. When he grown Streptococcus pneumonia on a culture plate, then he found two types of bacterial colonies. Some produces smooth shiny colonies called S strain and other produces rough colonies called as R strain. The smooth shiny colonies of S strain is due to a polysaccharide mucus coat which is absent in R strain. S strain bacteria are virulent, whereas R strain of bacteria are non-virulent. Means they do not causes any types of diseases. So, mice infected with the S strain die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Meanwhile, when Griffith injected a mixture of heat-killed S strain and live R bacteria, then the mice die. And he also recovered living S bacteria from the dead mice. After doing all these experiments, he concluded that the R strain bacteria had somehow been transformed by the heat-killed S strain bacteria. Some transforming principle transferred from the heat-killed S strain had enabled the R strain to develop or synthesize a smooth shiny polysaccharide coat and thus it become virulent. This is only due to the transfer of genetic material from heat-killed S strain to live R strain. This is the steps and result of Griffith experiment. When mouse injected with S strain, then the mouse died. Mouse infected with R strain, then mouse survived. Mouse injected with heat-killed S strain, mouse survived. But when mouse injected with heat-killed S strain and living R strain, mouse died and from its blood live S strain bacteria recovered. Mouse injected with heat-killed S strain and living R strain plus DNase enzyme, mouse survived. Why? Because DNase destroy the DNA. Mouse injected with heat killed S strain and living R strain plus protease enzyme, then mouse died of pneumonia. So by these experiments he found that the genetic material is DNA. This is the figure about Griffith experiments. It is shown here that when mice is infected with S strain, the mice die, and when mice is infected with R strain, the mice live, and when mice is infected with heat killed S, then mice live, and when mice is infected with a mixture of R live and heat killed S strain, then the mice die. So by doing this experiment, it has been proved that DNA is the genetic material. Now the next topic for today's discussion is biochemical characterization of transforming principle. Before the work of Oswald Avery, Colin MacLeod and Maclean McCarthy in the year 1933 to 44, it was thought that protein was the genetic material. These scientists worked to determine the biochemical nature of transforming principle of Griffith experiment. They purified biochemicals, that is proteins, DNA, RNA, from the heat killed S cells to confirm which one could transform live R cells into S cells. They discovered that DNA alone from S bacteria cause R bacteria to become transformed. They also found that protein digesting enzyme, that is protease, and RNA digesting enzyme, that is RNase, did not affect transformation. So the transforming substances was not a protein or RNA, it was DNA. Digestion with DNase inhibit transformation. So that confirms that DNA is responsible for transformation. So children, this is all about today's session. Thank you and have a nice day. For any kind of doubts, please write your questions in comment section.